Good afternoon, everyone. Welcome to our seminar series this afternoon, MHRC Seminar series. It's a pleasure for me to introduce Dr. Celine Aguer from McGill University, Outaouais branch. And she's going to be telling us about myokines. But let me tell you a little bit about Celine. She's originally from France, in the Alps region of France, just south of Marseille. And she did her PhD at the University of Montpellier, and then came to Canada and worked at the University of Ottawa with Mary Ellen Harper for 4 years as a postdoc, doing work on mitochondria and things like that in muscle and other tissues, and acquired a laboratory at Montfort Hospital in Ottawa, where she holds a lab and has had that lab since 2013. And she's still working at Montfort Hospital, but has this additional position with McGill University, where she teaches pre-med students in French in Gatineau, Quebec, twice a week. So big job and lots of work. And we're anxious to hear about Celine's work on autocrine role of myokines in regulating skeletal muscle metabolism. So Celine, welcome. And if you don't mind sharing your screen with us, and take it away, please.
So thank you for this nice introduction. I hope the screen sharing is still working.
It looks good.
Perfect, thank you. So thank you for this invitation. So today I will present some work of my lab on the autocrine role of myokines in the regulation of skeletal muscle metabolism in health and in the context of type 2 diabetes. I think I will choose the pointer, sorry, yeah. So I have no conflict of interest to declare. So as you are probably aware, the diabetes is reaching an epidemic level in the world. And there is around 11% of adults living with diabetes in North America. And it's supposed to increase to 13% in 2045. So the International Diabetes Federation showed that there is a high increase in diabetes cases in the world since 2000. And you can see that this increase was even more deep between 2009 and 2011. And they expect that 700 million of adults will live with type 2 diabetes in 2045. So right now, 1 in 11 adults live with diabetes in the world, which represent 463 million people. And importantly, 1 in 2 adults with diabetes are probably undiagnosed. So this number is probably much higher than what we think. And it's representing 10% of global health expenditure. So it's a huge problem in healthcare. So my lab, we are interested in the type 2 diabetes, so which represent 40% of the total cases of diabetes in general. And I will explain to you very quickly what is type 2 diabetes, and very simply, I will say. So when we are insulin sensitive, we can maintain a normal level of glycemia with a normal insulin secretion, as you can see here. But there is a diabetic state that occurs with insulin resistance. So insulin resistance, it's a decreased insulin action on insulin target tissues that are mainly the muscle, the adipose tissue, and the liver. And with insulin resistance, we can see a decreased glucose uptake and glycogen synthesis in the skeletal muscle, an increased lipolysis in the adipose tissue, and a decreased glucose uptake and increased glucose production by the liver. So in this state of insulin resistance, you can see that the pancreas compensate the insulin resistance by increasing insulin secretion. And this can allow to have normal glycemia level. Or whether with time, the pancreas become tired, which with a decreased secretion of insulin, which leads to a hyperglycemia. And it's at that point that we call that diabetes, or type 2 diabetes in this case. So my lab focuses on skeletal muscle. Why? It's because it's representing 40% of total body mass in a lean individual. And skeletal muscle, due to its mass, it's using a lot of glucose. And for example, it's representing 80% of insulin glucose disposal after a meal. And with type 2 diabetes, there are a lot of muscle disorders, especially at the level of metabolism. So there is a decreased mitochondrial function, decreased muscle fatty acid oxidation, and there is a development of insulin resistance. And this will decrease muscle function. So with a decreased fatigue resistance, a decreased force, and decreased muscle mass. So as a consequence, there is a reduced mobility and decreased level of physical activity with people living with type 2 diabetes. So this decreased physical activity will increase their mass gain, and so those people will become more obese. And as obesity is a high risk factor to develop insulin resistance, we are kind in a vicious cycle where those people with diabetes do less physical activity, which increase their insulin resistance, worsening their type 2 diabetes. So physical activity is well known to improve muscle energy metabolism, such as increased mitochondrial function and fatty acid oxidation, and increased muscle insulin sensitivity. And with sedentarity, this is exactly the opposite that occurs. So those are papers that were published on a bed rest study. So it's individual that stayed in a bed for a week or a couple of weeks, and they measure their mitochondrial function and insulin sensitivities, specifically in the muscle. And they found that being inactive decreased muscle mitochondrial function and muscle insulin sensitivity. And it's important to note that there is an increased sedentarity in individuals with type 2 diabetes. So this is possible that this decreased physical activity in individuals with type 2 diabetes result in these muscle metabolic alterations. So how physical activity increase the metabolic health and also the function of distant organs? So it's probably through different pathways, but in the last 20 years, it has been shown that muscle can produce small peptides or small proteins and act on distant tissues to improve their function. So during exercise, muscle contraction increase myokine secretion, and those myokines can act, for example, on the pancreas to improve insulin secretion. It can improve endothelial function. Some myokines can increase lipolysis and burning of white adipose tissue. Others are known to increase metabolism in the liver. And although we have some myokines that can improve brain function. Those myokines can also act locally at the level of the muscle, so it can be considered as an autocrine or a paracrine role of myokines. So it's known that some myokines increase hypertrophy, which can explain why exercise increase muscle hypertrophy. And other myokines are known to act on skeletal muscle metabolism. so improving insulin sensitivity and glucose uptake, fatty acid oxidation and mitochondrial function. And some myokines act through the AMPK pathway. And this pathway is known to improve fatty acid oxidation, mitochondrial function, glucose uptake. So it's possible that myokines improve AMPK activity, which in turn improve muscle metabolism. And importantly, myokine circulating levels are altered with type 2 diabetes. So it's known that, for example, the bad myokine, like the myokine that are more inflammatory myokines, are increased with diabetes, and the good myokines are decreased with diabetes. But it's not like white and black for sure. It's more complicated than that. So in my lab, we first focused on a specific myokine, which is interleukin 15 or IL-15. And this was the work of my first master's student, Lucien Nadeau. So we focused on that myokine because it was shown in the literature that IL-15 had an autocrine effect, increasing mitochondrial function and fatty acid oxidation in skeletal muscle. And the IL-15 was also shown to increase insulin sensitivity and muscle glucose uptake. And in IL-15 overexpressing mice, it was shown that IL-15 was increasing the switch from the glycolytic to oxidative muscle fibers. And as a consequence, the IL-15 overexpressing mice had a higher endurance capacity, a higher whole body energy expenditure. So they had lower fat content, which was probably one of the reason why they had an improved whole body insulin sensitivity. However, in those studies, they used like a level of IL-15 that is supraphysiological, so very high compared to the level of IL-15 secreted by the muscle in response to exercise. So Lucien decided to treat muscle cells with IL-15 that was representing the level of IL-15 secreted during exercise. And he first showed that basal glucose uptake was increased with 10 to 20 nanogram per ml of IL-15 treatment for 3 hours on L6 myotubes. So this is higher than what we find in circulation in response to exercise, but it has been estimated that it is the level of IL-15 in muscle interstitium, so locally at the level of skeletal muscle. And it's important to say that only basal glucose uptake was increased with IL-15 treatment. There was no improvement of muscle insulin sensitivity. Then Lucien measured mitochondrial function. So he measured resting mitochondrial function and the maximal capacity of the mitochondria using FCCP treatment. And it demonstrated that with very low level of IL-15, so this is the concentration we have in circulation after an exercise bout. And with those level of IL-15, he had a 40% increased mitochondrial function in the resting state and in the maximal capacity as well. So we wanted to better understand why IL-15 was increasing mitochondrial function. So we thought about the mitochondrial supercomplexes. So you probably know your electron transport chain that you learn in your undergrad class in biochemistry, where the electrons are transferred from one complex to the other to ultimately allow ATP production by ATP synthase. But it's not really working like that. We now know that complexes can form supercomplexes to be more efficient. So those electron transport chain supercomplexes contain complex 1 complex 3 and complex 4. There is a typo on this complex here from that paper. It's not 6, it's 4 for sure. And this assembly of complexes allows to have a more efficient electron transport chain. So with less reactive oxygen species production and higher ATP synthesis. And as you can see, complex 2 is alone, so it's not participating to the super complexes. So it was shown by one of the MD PhD students from Marianne Harper Laboratory in Ottawa. So it was Gadi Antoun who showed that the muscle of patients with type 2 diabetes, the white bar here, had lower levels of supercomplexes than the control subjects in black. So that's the case for supercomplexes containing complex 1, complex 3, and complex 4. And as I told you in the previous slide, complex 2 doesn't participate to supercomplexes, and it didn't find any differences. So apparently, muscle electron transport chain supercomplexes are decreased with type 2 diabetes. And in another paper from the Aguer lab in Lausanne, they showed that training elderly people for several weeks with endurance training, those people had higher muscle level of supercomplexes after the training. And this was linked to an improvement of mitochondrial function measured by oxygen consumption rate. So apparently, electron transport chain supercomplexes can be improved with exercise training. And we were wondering if IL-15, interleukin 15, can increase these levels of supercomplexes. So we collaborated with Dr. David Patton, who was at that point a postdoctoral fellow in the Harper lab. And so David measured the electron transport chain supercomplexes in L6 muscle cells treated with IL-15. And he found that there was a tendency for an increased complex 1 supercomplexes, as you can see on the blot here, with very low level of IL-15 that represent physiological levels of IL-15 after an exercise bout. And this was significant for complex 4 containing supercomplexes, as you can see here. So to summarize here in this paper published by Lucien Nadeau, who was my first master student, he demonstrated that IL-15 increased mitochondrial respiration. And this was probably due to the improvement in supercomplexes formation. He also showed that there was an increased glucose uptake, basal glucose uptake with IL-15 treatment. But we were unable to demonstrate that it was due to GLUT4 translocation. So it's probably due to another glucose transporter. And I didn't show you, but it also showed that IL-15 was increasing the phosphorylation of AMPK. So it's possible that IL-15 acts through the AMPK to increase mitochondrial respiration, supercomplexes formation, and glucose uptake. So now I will show you some data that we did on humans. So in collaboration with Lauren Sparks from Orlando. And those experiments were run by my current PhD student, Léa Garneau. So Lauren sent us some blood samples from women that did an acute exercise bout at 60% of VO2 peak. And we compared lean with women with obesity. And we had blood samples pre-post exercise and during the recovery period up to 24 hours. And Léa measured different myokines in circulation. So I have selected 4 of them here with interesting results. So she demonstrated that spark level was lower in women with obesity compared to the lean woman, as you can see here. Interleukin 8 level was also lower in the woman with obesity compared to the lean woman. And for FGF21, so fibroblast growth factor 21, she had interesting results showing that there was an interaction group time factors. So it means that, for example, as you can see, there was a tendency for the obese woman to have a higher FGF21 levels at rest and in response to the exercise bout. But during the recovery period, there was an increased FGF21 after 2 hours in the recovery period in the control group in black here. But this increase didn't appear in the obese group. And for interleukin 13, she had a group effect, meaning that interleukin 13 was overall higher in the woman with obesity in pink compared to the control woman. So I will talk later about the limitation of this type of study by measuring like myokines in circulation. But we also did a second study where we measured myokine circulation in collaboration with Jennifer Reed, who is a researcher at the Heart Institute in Ottawa. And she did that control trial where she had patients with coronary artery disease. And they compared 2 types of exercise training. So the first type of exercise training was classic endurance training protocol, so a moderate intensity continuous training with 30 minutes of training. So the total training session was 1 hour, but the individuals were training for 30 minutes at this heart rate, and they were training twice a week for probably 3 or 4 months. I don't remember exactly. And they were comparing this MICT with the high intensity interval training, so HIIT, where the individuals were doing 4 minutes at 85-95% of peak heart rate, and then 3 minutes at 60-70% of peak heart rate. And they were repeating that 4 times for a total of 28 minutes. So this training session was 45 minutes in total, and this one was 1 hour in total with the warm-up and cool-down. So Leah measured different myokines in the circulation of those individuals, and she compared people with diabetes. So there are the triangles on your right here, and without diabetes, which are the cycles here on your left. So for fibroblast growth factor 21, she found an interaction between type 2 diabetes and the effect of time. So what we call effect of time here is the effect of training independently of the type of training. So it's not comparing MICT to HIIT, it's just the training in general. So she found that there was a trend for higher levels of FGF21 in the patient with type 2 diabetes, and this was only decreased with HIIT in the diabetic group here, as you can see. Then for interleukin 8, she had an effect of time, so training effect independently of the type of training, and an effect of type 2 diabetes. So you can see that the levels of interleukin 8 is has a tendency to be higher in the diabetic group compared to the non-diabetic group. and there was a decreased level of IL-8 with the training, which was independent of the training type. For interleukin-6, there was an interaction between type 2 diabetes and the effect of time. So as you can see, interleukin-6 was decreased with training independently of the training type for the diabetic group, but it was not for the control group. And then for IL-10, so interleukin-10, there was a decrease with the training independently of the training type. And for IL-18, it was similar. So in general, we didn't find that HIT was better than MICT to decrease those inflammatory myokines, but we had some interesting results showing differences between the patients with diabetes compared to the individual that were not affected with the disease. So yeah, I told you a few slides before that there are like kind of limits measuring myokines in circulation, and it's because other tissues secrete similar peptides and similar proteins. So when there is an increased or decreased myokine secretion in response to exercise, it doesn't mean that it's coming from the muscle if we are measuring the protein or the peptide in the circulation. And another limit that I found in the literature, it was that most of the time myokines were studied like individually. So each like a myokine, it was the effect of a single myokine, and most of the time those studies were using supraphysiological concentrations of the myokine, so it's not representing what is happening in response to exercise in vivo. So how to counteract these limitations? So we thought that we can maybe use isolated muscle, because muscle so when they are isolated, if they secrete something, it's for sure coming from the muscle. However, it's possible to do that for sure with rodents or like with mice. We can isolate the soleus or the EDL, for example, and we can study that ex vivo. But with human muscle, it's more complicated because when we do a muscle biopsy, the muscle fibers are damaged. So if we put the muscle fibers ex vivo to measure the myokine secretion, I don't think we will see what's happening in vivo because the muscle fibers are damaged. So a way is to use the primary myotubes coming from those muscle fibers. So what are those cells? They are the satellite cells that are at the periphery of the muscle fiber, and they can be activated to proliferate into myoblasts and differentiate into multinucleated myotubes. And in vivo, they can become muscle fibers to repair the muscle when there is an injury, for example. In vitro, we stop at the level of myotube. So they are not mature muscle fibers, they are the step before. So we double check if the cells express desmin, which is a muscle marker. And we use the cells only if they are 90-95% desmin positive. And we can also check if they express differentiation markers such as troponin T, which is only expressed in myotubes and not in myoblasts. And as you can see here, we have nice muscle fibers, multinucleated muscle fiber, I said muscle fibers, it's not muscle fibers, multinucleated myotubes, sorry. So is it a good model to measure myokine secretion and muscle metabolism? So it has been demonstrated by others that when those myotubes are from trained individuals, they maintain in vitro their higher mitochondrial capacity So you can see here that they have the trained myotubes, or the myotube from the trained individual, I should say, have higher oxygen consumption rate, which was significant with FCCP. So it's a maximal oxygen consumption rate here. They also had higher levels of protein, of mitochondrial protein, as you can see here, compared to the untrained individuals. And those myotubes also had increased succinate dehydrogenase activity, succinate dehydrogenase being enzyme in the Krebs cycle and the electron transport chain. This paper from Cédric Moreau in France showed that individuals with obesity that were trained for a few months, they had a higher glucose oxidation and higher glycogen synthesis in their muscle, and it was the same in their myotubes coming from the muscle. So you can see in white here is pre-training, in black post-training, and they had increased glucose oxidation after the training. They also had increased glycogen synthesis after the training, and this increased glycogen synthesis was even more pronounced in presence of insulin, showing higher insulin sensitivity in the myotubes from trained people. And with type 2 diabetes, it's well known that the myotubes derived from the muscle of people with diabetes, they maintain in vitro their metabolic defects. So it was shown, I think the first paper showing that was a paper from the Henry lab in San Diego. And they showed that insulin resistance was maintained in those myotubes from type 2 diabetic people. And after that, a lot of papers from the Gaster lab was showing that. So decreased glucose uptake, insulin stimulating glycogen synthesis, for example. And during my training, we also showed that those myotubes, so also maintain some energy metabolic defects. So here they were not from people with diabetes, but people that were insulin resistant and obese. And we showed that fatty acid oxidation was decreased in the myotube of the obese individuals compared to the lean control. And we also showed that they had a lower metabolic flexibility. So what is metabolic flexibility? Here it was the capacity of the myotube to inhibit fatty acid oxidation when they were put in presence of glucose. So you can see in the lean myotubes, the presence of glucose was inhibiting fatty acid oxidation almost by 75–80%. But it was decreased to 50% in the myotubes from the obese group. And we also showed that mitochondrial function, so oxygen consumption rate, was lower in myotubes from people with diabetes. And this was only the case when the cells were cultured in a galactose medium. So when they are in like glucose medium, those cells are are highly glycolytic, and so they don't use a lot their mitochondria. So to push the cells to use their mitochondria to produce ATP, we put them in a galactose medium. And what is interesting with galactose is that glycolysis of galactose doesn't yield any ATP. So at the end of glycolysis, when you have the pyruvate has to enter the mitochondria to produce ATP. There is no ATP produced during the glycolysis of galactose. So it's pushing the cells to use mitochondria, and we can detect with this model mitochondrial dysfunction in myotubes from diabetic people. So the Henry lab in San Diego also showed that the myotubes from individuals with diabetes had altered myokine secretion, and it was most of the time an increased inflammatory myokines. So they maintained their altered myokine secretion in vitro. So we decided to use that model and to have in vitro model of exercise, which is so electrical pulse stimulation that I will call EPS. And this stimulation can increase, I think I have to stop that to show you the video. So it's increasing myotube contraction. So you can look here, you can see the contraction. You can see the contraction here. So maybe I will show you again that the cells are contracting with the EPS. So we also looked at markers of muscle contraction, such as the phosphorylation of acetyl-CoA carboxylase, which was nicely increased with EPS in black here, and membrane GLUT4, which was also, so the translocation of GLUT4 was also increased with EPS. So this work was done by Hannah Khoury during her honors project last year, but Hannah is now a master student in my lab. And we also showed that EPS was increasing interleukin-6 secretion. So interleukin-6 is the most studied myokine. So this work was done by Leah. So using this model, we would like to determine if physical exercise can increase myokine secretion, which in turn can improve mitochondrial function and insulin sensitivity. And the opposite will be that with diabetes, there is a negative alteration of myokine secretion that can explain mitochondrial dysfunction and insulin resistance. So I will now show you very new data from our lab. So they are preliminary data. So the first experiment we did was to treat the myotubes from healthy participants with or without EPS, and we took the condition medium that contained the myokines, but also other factors secreted by the myotubes during contraction. And we put this condition medium on healthy myotubes, but that were not stimulated with EPS. So I call them resting myotubes. And we looked at insulin sensitivity. So here you have the control condition. It's myotubes in their differentiation medium. And you can see the increased insulin stimulated glucose uptake. So the black bars are with insulin, the white bars are without insulin. So when we took the medium, the condition medium without stimulation, so NS is not stimulated, we already saw an increased glucose uptake in the basal state, which was still increased with in the insulin state. So it's important to tell you that myokines are not only secreted during muscle contraction, they are also secreted at rest. So apparently there are some factors, maybe it's myokine, maybe not, we don't know because we didn't measure them or inhibited them in that study. But there are some factors secreted at rest by muscle cells that can improve basal glucose uptake and insulin stimulated glucose uptake. After 1 hour of EPS or 24 hours of EPS, so the condition medium was increasing even more glucose uptake. So keep in mind that it's an experiment that was done only on one set of myotubes, so we have to reproduce the data for sure. And Leah also looked at mitochondrial function, and she looked at resting oxygen consumption rate and maximal oxygen consumption rate. And here, so she did the same experiment where she took the condition medium of non-stimulated or EPS-stimulated myotubes, and she put them on resting myotubes. And so this is the condition medium of non-stimulated myotubes and with EPS stimulation. And you can see that there is something in the condition medium that is increasing mitochondrial function in the basal state. and maximal mitochondrial function here. So then what she did, the two other bars here, she precipitated the proteins from the conditioned medium. And since myokines are protein, they should be in the precipitated proteins. And she resuspended that in fresh medium. And you can see that she had still an increase with the conditioned medium, or the protein, the precipitated protein from the conditioned medium resuspended in fresh medium. She still had this improvement in resting mitochondrial function. So it's likely that this increase in mitochondrial function is due to the myokines that are secreted during EPS stimulation. So for the second part of the study, we are looking at the negative effect of the myokines secreted by the muscle of patients with type 2 diabetes, and to determine if this altered myokine secretion can explain why people with diabetes have altered muscle metabolism. So here, Leah took the conditioned medium of myotubes from patients with diabetes, and she put that conditioned medium on healthy myotubes to see if something in the conditioned medium can alter muscle metabolism in healthy myotubes. So this idea came from one of my PhD paper, where we did that experiment, and we showed that the conditioned medium of a diabetic subject was increasing the level of a fatty acid transporter, FAT-CD36, at the plasma membrane, which is known to increase lipid accumulation in skeletal muscle in patients with type 2 diabetes. So you can see that there is almost no expression of this fatty acid transporter when the control cells were incubating with the control medium. But when they were incubating with the conditioned medium of type 2 diabetic patients, there was a nice increase in FAT-CD36 expression. So Leah first measured the levels of myokines in the conditioned medium of those myotubes. So she had myotubes from one healthy participant, one obese participant, and one patient with type 2 diabetes. I know that it's only an N of 1, but the subjects are very well matched on sex. They are women, on age, and we also match them on their physical activity level, so which was measured by accelerometry. So you can see that for the age here, the healthy participants, they had an increased IL-6 secretion with the EPS. So the EPS is in black, and the resting condition, no EPS, is in white. So we have this increase for IL-8 as well, for IL-10. There is nothing happening for FGF21, and we have a nice increase with for SPARC as well. And we also have myotubes from one obese participant that is so much with the BMI with the diabetic person. And as you can see, there is a tendency for an increase in those myokines from the obese participant. Again, nothing for FGF21, but for the myotubes from the type 2 diabetes participants, you can see that they secrete a lot of those inflammatory myokines. So we confirm with our model that myokine secretion is altered with diabetes. So here, what Leah and Hannah did, they took the conditioned medium, so containing the myokines of patients with type 2 diabetes, and they put that conditioned medium on healthy myotubes. And the control condition here was the conditioned medium of a control myotubes onto control myotubes. they measured glucose uptake in response to insulin. So here it's the insulin stimulated glucose uptake that is presented on this figure. So the white bar are the control. You can see that they respond to insulin. And the black bar are when the control cells were exposed to the conditioned medium of a patient with type 2 diabetes. And you can see that for these two control cells, incubating them with the conditioned medium of diabetic myotubes decreased their response to insulin. Hannah also did experiment to look at the insulin signaling pathway. So she looked at phospho-AKT, which is central in this pathway, phospho-GSK3, which is involved in glycogen synthesis, and phospho-AS160, which is involved in GLUT4 translocation. So in the control cells exposed to the control medium, she found a nice increase in the phosphorylation of those proteins, as you can see here, in presence of insulin compared to the basal. But when the control cells were incubated with the conditioned medium of the diabetic myotubes, she had a decreased response to insulin, as you can see here. So what was interesting with the first figure here is that, as you can probably tell, some conditioned medium of diabetic patients were not decreasing insulin sensitivity. This is the case, for example, for this patient, which we label D. And I was less wondering why this is the case. And I looked at the characteristics of the participants. And interestingly, this specific patient had the lowest HbA1c level. So HbA1c is a marker of glycemic control in the past 3 months. So the lower HbA1c is, the better the glycemic control is. So having a low glycemic control is good. A low glycemic control, sorry, a low HbA1c is good. So it's interesting to see that the patient who has the better glycemic control have the lower effect on the control myotubes. So I was wondering if there is a correlation between these two parameters. And interestingly, so I took the data from this figure on the x-axis here. So this is exactly the same data, insulin stimulated glucose uptake. And I plotted them against HbA1c levels, and it was negatively correlated. So which means that the conditioned medium that is giving inducing the most insulin resistance in the myotubes are coming from the patient with the lower glycemic control or the higher HbA1c level. So to conclude, we have some data showing that it's possible that the muscle secretome is altered with type 2 diabetes, and it may participate to the muscle metabolic defects seen in the muscle of patients with diabetes. So it's maybe due to an alteration in myokines, but it's important to tell you that muscle cells don't secrete only myokines. They also secrete metabolites, microRNA. So it's possible that it's coming from other factors secreted by muscle cells. And we will have to determine that in the future. So I would like to acknowledge all the people who participated in those studies. So this is a recent picture of my lab. We were allowed to go to the restaurant. It was 2 weeks ago. So it was to celebrate Christmas in March. And so the work shown in that presentation was mainly done by Léa Garneau, my PhD student, and Hannah Coury, a master student. And the first part of the presentation on interleukin-15 was done by Lucien Nadeau, who was a master student in my lab. And I would like to thank the collaborators and answer and Société Francophone du Diabète, which are funding my research. Thank you. If you have any questions, I will be happy to answer them.
Celine, thank you very much. Very nice. Very nice.
Oh, wow, it was long, sorry.
Oh, no, you're good. You're good. We have time for a few questions. It's okay. I remind the viewers, please, to type your question into the Q&A forum there, and I'll read them, and then Celine can answer them. So, you know, I'm not in this field at all about interleukins, and part of the problem for me is that I don't know which ones are the good ones and which ones are the bad ones. So I really have trouble with this. So tell me, you know, IL-15, for example, there was a nice paper in Science or Nature or something like that some time ago that showed increase in mitochondria when you have, isn't it activating the receptor and going through a pathway in which mitochondrial content goes up. I think you showed in your first set of experiments here with IL-15 that, I don't know whether it was in C2, C12 myotubes that you did it, but didn't you show an increase in mitochondrial content, increase in glucose uptake? What was the model you used there?
It was L6 myotubes.
Ah.
So they are muscle cells from, like immortalized muscle cells from rats.
Right. Yes, and the L6 cells, right. So it seemed to me that you showed some beneficial effects of IL-15 on metabolism, right? And then later I thought you showed that IL-15 is secreted to a greater degree in diabetic, you know.
So yeah, it's what I said at one point. It's not like completely like black and white for sure. The thing is, apparently, so I don't think it was proven for IL-15, but it was for IL-6 and other myokines that the muscle of patients with diabetes have some myokine resistance.
Oh.
So they don't respond normally to myokine treatment, for example.
I see. Okay.
So it's not the case for all the myokines, but for some of them it is.
Yeah.
So it's possible that they have increased secretion of some myokines that are supposed to be good because they don't respond well to those myokines.
Mm-hmm. Yeah. So let me go back to that IL-15. You showed early on that it increases glucose uptake in muscle, increases mitochondria, increases respiration. And yet it seems to be, I think your last slide, you said that these myokines, and I don't know if IL-15 was included in that, reduces insulin sensitivity. But that seems at odds with the experiments that you showed in the L6 cell. So I'm a little confused about whether IL-15, for example, is good.
Yeah, it's again, so it's well known for IL-6. If you have like a certain level of IL-6, it's beneficial, but if it's too high, and chronically too high, it's inducing insulin resistance. So that's probably what's happening.
I see.
So acute level of one myokine or several myokines in response to exercise can be beneficial, but when it's very high level of those myokines, it can be bad. Detrimental,
sure. Yeah. So the changes that we see with exercise that you showed in your 2 human studies with training or with exercise, these are transient changes in these myokines, I guess, that might have a beneficial.
So the first human study, it was the effect of an acute bout of exercise, so it's transient for sure. The second study, it was the myokines were measured at rest. So the individual were not training for at least 24 or 48 hours before taking the blood samples. So it was, it was after I think it was 3 or 4 months of training, but it was measured at rest. It was not measured in response to an acute bout of exercise.
Okay. Right. Training seems to reduce. Am I correct? Training reduces the levels of these.
Yeah, it's what we found. Yeah, that for most of the myokines, training is reducing the myokine level, but at rest again. It's not in response to an acute bout of exercise. What will be interesting is to see if training affect the acute response, to exercise in terms of myokine levels.
Yeah, it's like the insulin response to training. Insulin levels at rest are lower in a person that's trained. So same thing, I guess. Yeah.
It's
Okay.
it's a lot of things are very similar between insulin and myokines, I can tell.
Yeah. Okay. That's helpful. That helps me.
To understand.
Okay. Ola asks, thank you for a very informative presentation. Given that some of the myokines that were increased by exercise or electrical stimulation were also increased in diabetic patients, do you want to comment on the functions of these myokines in health versus disease? And did you follow the question?
Yeah. But I think it's something that I already answered you before. Yeah. It's
maybe,
kind of, like an acute increase in myokine can be beneficial for the muscle. But when it's chronically increased, I think, my opinion, it's not proven, but my opinion is it's probably not good. Yeah. Okay.
Ola had the same question I did. In your 2 human studies, you didn't seem to follow IL-15. You had a bunch of other markers, but your original studies, you were talking about IL-15, but
I didn't show. We did IL-15, yes, but we didn't have any response at the level of IL-15. That's why I didn't show it.
Okay.
So we didn't have differences between the groups. Yeah.
Okay. Adam,
IL-15, it's in the literature, it's very, some labs, they showed increased IL-15 with diabetes or obesity, and others, they showed decreased. So it's hard to say. It's very, it's not clear at all.
Okay. So
and we found no differences. So I don't know.
Yeah. No, it's clearly a difficult field, and the variability among subjects is really, it's difficult. So Adam is asking to clarify the state and the intensities of the training that you used in the training studies you described. But what were the intensities of the training?
Which one? Because there are 2 studies. The first one,
you had a moderate intensity continuous, and you had a hip training one, I think, right?
Yeah. So the moderate intensity, the MICT, they were training twice a week at I think it was 20 to 40 heart rate, bit higher than resting heart rate, if I'm saying that well in English, sorry. And the high intensity interval training, it was 85 to 95% of their heart rate peak. So it was very intense. Right.
Okay, good.
So we didn't train the participant. It was samples from Jennifer Reed at the Heart Institute in Ottawa. So she had like a whole clinical trial with comparing different type of training. So we just collaborated with her and analyzed the samples.
Yeah, right. Okay. Daniel is asking, in adipose tissue, FGF21 exerts beneficial effects like browning. In muscle, it appears to be decreased with HIIT training. How does FGF21 influence muscle? This is question.
That's a good question. I don't remember this one in particular. FGF21, I think, I don't want to say anything wrong. I'm sorry.
That's
I don't remember by heart.
That's okay. Let's move on. Jeff Brault says, nice work in your cell studies. Do you think IL-15 activates AMP kinase directly, or does it increase energy demand, thus activating AMPK indirectly?
Yeah. So we measured in that study, we measured ATP, AMP ATP levels, and it was not altered with IL-15 treatment. So it's probably not due to decreased ATP production or something like that. We also looked at STAT3, which was in our hand, it was not like the phosphorylation was not increased with IL-15, but other labs showed that it was increased, but they were using higher level of IL-15 that we did. So maybe that's the reason why we had different results.
Right, right, right. So if you had to pick an interleukin or myokine to follow, and you're going to go down this path, which one would it be? Which
That's
one's
a good
gonna...
question.
Yeah.
I think, right now I don't know, to be honest, because in the literature, like the favorite myokine is irisin, but there are a lot of discrepancies around that. We know a lot about IL-6 because it has been well studied,
Right.
especially by Benke Pedersen. Yeah. And but right now I'm not sure. I think I will want first to see in our model which ones are the most affected by diabetes, and to see, because we are planning to do functional studies to inhibit one or the other myokines, to see, like, from the condition medium, to see which one is involved in those metabolic defects, for example. Yeah. So I think I would like to do that first and then choose the myokine to work on. Yeah. You could always inhibit selectively in the tissues that you're applying the conditioned media to. You could always selectively inhibit. It'd be a shotgun approach, but you could inhibit the receptor, knock it down for IL-15, for example, using siRNA, or if there was a drug, a specific agent that blocked the receptor, then you could investigate them individually. But I guess that would be a lot of work. If you it will be a lot of work.
Yeah. Right.
That's why I need my CHR grant.
Indeed, that's what we all need. Right. Well, it's almost 5:04, so I think we better stop there. You've had this was very, very good, Celine. Thank you so much. Just stay with me for 1 minute, please.
Thank you. Thank you.
And dear audience, thank you very much for being with us today. We'll see you at the next seminar. Our seminar is over now, and we'll see you the next time. Very good. There we go. All right. That's excellent. Well, as I said, I think we have not had a myokine talk before, so I really enjoyed that. That was good.
Okay, cool. Thank you.
Many studies, and clearly you're on the right path for investigating this. I really like your conditioned media type experiments, and
I always was it clear what we did exactly, because I always think that it's hard to explain. So I hope it was clear to everyone. Okay.
That was clear. I mean, it was clear to me. I thought it was. I think it's a really nice approach, where I haven't done that yet myself with conditioned media, but I really like it. I like the idea. We use the same stimulator as you do. We have the EPS thing. We also have one that we made a long time ago, homemade one from the university electronic shop here that I actually find it's better, but it's old. And so I bought the same system that you have, it still doesn't work as well as my old system. So I go back and forth. Okay. But a lot of people use it to study myokines, don't they? Like there's so it's working well for that purpose. I like to look at see if it increases mitochondrial content, and it's a touch and go. It doesn't. The contractions don't always produce an increase in mitochondria, at least in my lab. But
I don't know. It's so we try to use a 1 hour EPS protocol, so it's more like an acute bout of exercise. Yeah. And because I had some reviewers telling me that 24 hours or 48 hours, it's not representative of what a human does, So we didn't, we looked at sometimes at 24 hours EPS, but we didn't look into like a long EPS period. So 1 hour for sure. I don't think it will increase mitochondrial. No,
it won't. Yeah. I usually do. We usually use 3 hours a day, and then we give it a muscle cells a rest, and then we stimulate again and again and again, for four days. And in the old system that we had, we were able to see an increase in mitochondria, but not so much with this system.
Oh, okay.
Yeah, I don't know.
So do you know why, or?
No, I don't know why. I don't know why. I've had several people try it, but I don't know. Maybe it's the cell type. I use C2, C12 cells mostly. You use primary cells mostly, I guess, do you?
Yeah. It's mostly primary cell that we use with this model.
Yeah. Maybe it's my cell type. You don't use the C2, C12. You use just the L6 or the L.
We use the C2, C12 as well. The L6 cells we use them more like when we look at insulin sensitivity, because the C2, C12 are not very good to increase glucose uptake in response to insulin. But I have a student, honor student, who is doing EPS on C2, C12 and L6 cells. It does the two. So we didn't look at, but again, it does 1 hour EPS treatment. So
yeah,
I cannot look at mito content for sure.
No, of course. Yeah. Amira Clip would contend that the L6 cells don't contract. So you might find a difficulty with the EPS to make them contract.
So I think you're right. I'm not 100% sure, but I think my student told me that the C2, C12 are contracted, but not the L6.
That's right. Yeah.
So
yeah, I would not try. I wouldn't bother with the L6 because they have a long history of not being able to contract. They're great for the glucose uptake part, but not. And Amira Clip will use hypoxia. She'll use different drugs. But she never makes the muscle cells, tries to make the muscle cells contract. The L6 ones, they just don't do it. They don't have enough. They don't have enough cross bridges. Actin and myosin doesn't develop well enough, I think.
Yeah.
That's what they tell me.
Yeah, I think, and I think the C2, C12 are even better than the primary myotubes. Because we saw in the literature that there are like some labs, they are doing co-culture of C2, C12 with human myotubes, because apparently it's improving the response to EPS, and the C2, C12 can fuse with human myotubes. I don't know if you saw those papers.
I did not.
Yeah, they have so it's a co-culture. They mix C2, C12 and human myotubes, and they demonstrated that they can fuse together. It's improving their response to EPS.
Oh, okay.
Yeah.
Okay, well, I'll look for that. Now, my last question is, are you coming to IBEC? Are you going to come to this conference in Toronto in May?
I will, with my PhD student, Leah. She's supposed to submit an abstract soon. I think the deadline is next week or something like that.
1 week from today.
Okay, so she will do it last minute, because she's like that, always last minute. I don't like that, but I have to.
I know.
Yeah, and so we are planning to come in May.
Excellent.
So I will see you for sure. Yeah.
Beautiful.
And she will probably present like similar data, because it's all of our work on the human myotubes. That's fine.
Good. Well, I'm going to let you go. It's Friday afternoon. You're going to want to go and relax now, and so am I. So
Thank you. Have a good weekend.
Thank you very much, and same to you. And thank you for doing such a great seminar. Thank you very much.
Thank you for the invitation.
You're welcome. Okay,
and
we'll see you
in May. Bye-bye.
Yes. Bye-bye.